Hello, everyone. So today we're going to continue with our little discussion of the nervous system and its subsequent parts. So for now, let's go over a bit of the nervous system's organization. Now, neurons in the nervous system are organized in specialized things called neural circuits. So a neural circuit, you can imagine, is that let's take part of your spinal cord like this. Generally happens that there's an order of how things are. You'll get a sensory from the outside environment. You'll feel senses, and then that'll pass on to your brain, or whatever. It just starts entering your spinal cord, and then you can have voluntary actions or reflexes that goes towards what's happening outside. These connections can be local, which means it's within this region, or they can be projections to distant regions, such as the brain. Now, neuron circuits can be wired together. You can have convergence. This is where neurons can work together and input one signal to a mega neuron that goes to the brain. You can also have divergence, where one presynaptic neuron shares its input with multiple neurons, which also has ways of moving. And you can also have parallel processing, of which many neurons send the signal. I'll say parallel processing, where many neurons receive and pass on the same signal to different places. They can be wired together for excitation, like so, or they can also be wired together for inhibition, inhibit this part. This is what we can call recurrence. Now, to be more specific, the CNS has a majority of neurons and synapses in the body and also has additional areas for protection. So the CNS majority of are all the neuron cell bodies, and they're located in the skull and the vertebral column. If you want to imagine it's this sort of shape here. This is your brain, and then you have your spine like this. And intuitively, there's something called CFS or cerebral spinal fluid, cerebral spinal fluid that circulates from the ventricles in the subarachnoid space. So we have ventricles, which are supposed to be around here, and they sort of line up the subarachnoid space, which cushions the brain and also helps clean the brain from any sort of other issues or anything. Now, it cushions the brain by reducing impact. Let's say imagine a kind of like a water balloon. By having a water balloon like this, you can kind of just, if your brain is moving against your skull, well, it's not going to have as much damage. It also helps drain fluid from some sort of pumping. However, there's something called the blood-brain barrier. I'm going to call it the BBB. It resists the transport of water and molecules between the blood and the CNS. So let's say we have a CNS and blood here. It's a tight epithelium. So if you remember what we talked about with tight epitheliums, there's no paracellular transport. So this means that only lipophilic molecules or uh, hydrophilic with transport proteins can make it to the CNS. So it's very selective in what it brings. And also, astrocyte. Remember what we talked about earlier? Astrocyte control blood flow. Blood vessel, I think, yeah, vessel diameter, blood flow essentially in CNS. A functional MRI is one way of measuring blood flow under different conditions. The CNS also has forms of spinal cord and other ways of communicating with the brain and with the other systems. So the spinal cord is basically the destination of most afferent neurons of the PNS. Now, the spinal cord is first organized into four parts. I want to think of it as parts like this. You have the cervical part, you have the thoracic part, you have the lumbar and sacral areas of the body, and they're organized in these four areas. Now, Each spinal cord segment contains the same organization but processes information from different parts of the body. So let's say you'll have something from the cervical region. Your cervical region is around your neck. So if you have something in your neck region, well, it's going to process something that's more for your upper limbs or neck or anything. So as a rule of thumb, the cervical area, let's just say it's for upper limbs. Thoracic would be for your trunk. Lumbar is lower limbs. And sacral is lower limb, or more specifically, genital region. Within each part of the spinal cord, you essentially have a dorsal root ganglia, I'm going to call it DRG, ventral root, a dorsal root, and a spinal nerve. So how it works is these spinal nerves, they contribute to, at the very least, what should we call it? Sort of processing information towards the brain or towards the CNS. No, okay. Now, one thing you need to know is that when you sever the spinal nerve, you're basically saying no reflexes are going to happen. If you sever the dorsal root, what you're saying is no sensation is going to happen. And same thing in the ventral root. If you sever it, you're basically saying it's not going to have anything there. But let's say you just pinch it. If you pinch the ventral root, well, that means that it might still move something, but it's going to be paralyzed in a way. Now, remember what we said? The ventral horn, well, we also have horns that they attach to. The dorsal horn leads to sensory processing, and ventral horn also leads to is filled with motor neurons. Circuits contained within gray matter, which is the dorsal horn and the ventral horn, lead to reflexes. So what that means is, let's say we have a spinal cord like this. These are involved in reflexes. So if we're not talking about white matter, we're only talking about gray matter here. If if we lose the dorsal horn, or if the dorsal horn is damaged, that means that we're not going to get any reflexes because your body isn't able to pick up on any sort of outside environment. If you damage the ventral horn, what's interesting is, well, you aren't gonna express any reflexes, but you're able to kind of feel. You have no issues with any sort of sensory information. But at the same time, you can also voluntarily control the body. Now, similarly, you have two areas here full of white matter. This is called the dorsal column, and this is called the lateral column. And the dorsal column is the ascending sensory relay, and the lateral column is a descending motor commands. So these are for conscious commands. So you'll have a lot of questions dealing with that. Uh, what else? You also have a bunch of cortical regions of the brain. You have the frontal lobe, the parietal lobe, the occipital, and the temporal. Frontal, parietal, occipital, temporal. And within them, you also have the gustatory cortex, the primary motor, which we call M1, primary somatosensory, S2. You also have the auditory, olfactory, whatnot here, O1, A1, and the visually V1. What you have to know is the M1 and the S1. So M1, um, the primary motor cortex. This is where you have descending motor signals. In the S1, the primary somato uh, sensory cortex. So where you have the ascending projections. Well, I guess we can call them descending, ascending projections. And what's something interesting we can mention? Mm, I guess one thing that's kind of key here is, well, they decousinate for these signals, meaning that the M1 on the right side of your well, let's say on your brain, it's on the on the right side. You'll notice that they'll actually cross over and they'll affect the left side of your body. So if you're unable to control like the left side of your body, that means you have an issue on the M1 projection and you might have an issue on the M1 area in the right side of your body. And also, after they decussinate, you have to understand that there are differences. Let's see. Spinal motor neurons. M1 goes to spinal motor neurons, whereas S1 goes to dorsal roots. They have the relay synapse on the thalamus for S1, and then relays onto the hindbrain. Well, no, it will be first in the hindbrain and then the thalamus. And yeah, brain is lateralized and stuff in this case. EEGs are also one way of using electrical fieldograms through scalps, and you can detect seizures from this. And yeah, there's not that much to talk about for the organization aside from this. Another thing we can talk about is some senses that you might experience. I hope you're all able-bodied individuals with the ability to perceive things in your environment. If not, that's all right too. There's still ways. So, sensory or sensation, it means the detection of stimuli. And perception means awareness and meaning. Sensory is always induction. Perception you can call it as cortical activity, whereas sensory pathways are transducing information. Now, stimulus intensity is usually encoded with action potentials, and stimulus duration is also like, let's say you experience pain or you experience a sensation. These are also usually detected in the action potentials. Now, there's two sorts of now timing is really important, I should say. You can have phasic or rapidly adapting um, afferents. For instance, for the action potentials of rapid activity, they'll understand there's a sensation for these are all action potentials, and then they'll have none. And then in terms of the graded potential for this, it'll look kind of like this. So graded potential. However, the stimulus intensity is still going to remain like so. Perhaps this is where the stimulus is actually occurring, let's say. And the frequency of action potentials will be like, let's say they'll be increased at this area, and then increase in this area, and then they'll be gone. However, you can also have tonic or slow adapting. So how I think of it is TSA, tonic slow adapting, and this little action potentials, they'll fire all the way, all the time during the actual activity, I should say. The graded potentials are like this. And then let's say the activity or whatever the sensation is like this, you'll get some sort of block like this. So these are encoding the entire stimulus duration, whereas phasics only encode the stimulus onset. Now, the location of stimulus is usually based off the receptive fields of the specific sensory cells. For example, let's say you're touching a piece of skin, you're either here or here. Now, specific sensory cells are going to understand that it's going to be either here or there by the specific neurons in those locations. And these are called, you can call these the receptive fields, the fields where we feel something, where we feel receptions. Now, acuity which you can describe it as accuracy, is inversely proportional to 1 over receptive field size, and directly proportional to receptor density. So you have more of these, you'll more receptors, you'll understand like where it is more precisely. But if you have them in a smaller area, more dense, then obviously you'll also feel it more specifically. Receptor cells also have different sensitivities to stimuli, and yeah, there's also functional ideas that you can understand. Thermo sort of functional classifications. The most simple functional classification is usually they group nociceptors and thermoreceptors. These are pain and temperature, and the mechanoreceptors, which should target movement. Now, touch and mechanical and proprioception are from ion mechanically gated ion channels. And this means that if there's some sort of touch or sensation, they're gonna activate action potentials and lead to changes and senses. One example is that in tactile receptors, you have Merkel cells or epithelial sensory cells, and you have sensory neurons, whereas proprioceptors, which measure positioning and balance, you have muscle spindles and Golgi tendon organs. Now, different mechanoreceptors are sensitive to different types of stimuli because of their location and cell temperature and structure. So I guess some simple ideas are, well, you can have displacement of hair shafts. You can Merkel cells, tactile, I'll say tactile or Merkel are for pressure and texture. I guess one way to know is like Merkel has like the word itself has some textures. Thermoreceptors and nociceptors, thermo and pain, are from nerve endings. You have bulbous corpuscles for skin. Bulbous, I think a bulb is something that's really fat and big. Maybe you can think of skin. Vibrations are from lamellar. I guess a good idea is like, you know, the basketball player, LaMelo Ball, he moves a lot, and I guess movement is related to vibrations. I guess that's a way of remembering. And you also have some dendritic dendrites and tactile corpuscles for fine textures. Now, one thing that's key as well is that for sensory neurons related to pain and temperature, you have different stimuli ranges because they have different receptor proteins. Now, for thermoreceptors, they use TRPM5 and 4 and 5, I should say. So I'm going to draw a fire here. And then for pain, I'm going to draw like cross skull and crossbones. You have TRP V1 and TRP V2. And the thing that you should understand is that you feel thermoreceptors quicker, fast, more, you feel the onset of them um, quicker than they're more sensitive. You're more sensitive to heat than you are to pain, which kind of makes sense if you think intuitively. Like let's say you put your hand on a pan, maybe initially it's gonna be kind of warm, lukewarm kind of feeling. Maybe it won't feel that bad, but after a while it starts heating up. It gets really, really hot, and then yeah, that doesn't feel so good. And the main distinction is due to the afferent fibers actually between these afferent fibers. They just have different axon fibers, and that leads to different conduction velocities. So proprioception and touch, well, no, I mean, I should compare them to, well, I should compare to all the types, but whether or not they're myelinated has a very big difference. And these distinctions are maintained through spinal processing and ascending pathways. So what do I mean by this? So in touch and proprioception, they are maintained, the axons in the brain from the dorsal column pathway, they decussinate in the medial gracili fasciculus and lateral cuneate Fasciculus. They decussinate at the medulla oblongata. Whereas the thermoreceptors and nociceptors, they decussinate at the spinal cord. So there's different ways, I suppose. These pathways can sometimes interact though with some of them with sometimes, let's say, if you rub your body after you feel pain, the pain will feel less and such. Now, two other senses we need to talk about are involve olfaction and gustation. Olfaction relies on olfactory sense neurons in the nasal epithelium. So first you'll have these odorants that enter the nasal epithelium, and then they hit these olfactory sense neurons, sensory neurons, or these olfactory receptor proteins, which are metabotropic receptors, which leads to degraded potentials, degraded potentials that cause sodium entering. Uh, well, I guess I should talk about the steps. The odorant molecule is attached to sensory olfactory olfactory receptor protein. And then this this activates adenylyl cyclase, converting ATP to cyclic AMP, which activates a sodium ion channel, leading sodium to enter. And this causes a graded potential, where then information is passed to CNS. So the main players, I'll repeat it again. You have the sensory, you have the odorant that attaches to an olfactory sense neuron, and that activates adenyl cyclase, which converts an enzyme that converts ATP to cyclic AMP, which leads to sodium entering the plasma membrane, leading it to depolarize for a greater potential. And then if a significant depolarization occurs, the action potential, I should say, action potential is triggering the axon of which information is relayed to the central nervous system. And this passes directly to the brain, no brain stem or thalamus. Each olfactory sense neuron also only makes uh, uh, one odorant protein, I should say. So this way, they're all very unique in this sense. Gustation is based off five qualities and is from taste receptors and taste buds. And so the taste buds can detect multiple different qualities on your tongue, let's say. And then each taste uses specific transduction mechanisms and distinct pathway afferent neurons. And taste information enters the CNS via the hindbrain and then relays to the cerebrum via the thalamus. Olfactory intestation, and I think there's maybe one more we can talk about. Yeah, we can talk about sight and sound. So in the case of sight and sound, vision is mediated by eyes, which are very complex. You have the cornea here, you have your lens, cornea, you have your lens, and you have the anterior cavity with aqueous humor, and then you have the posterior cavity containing vitreous humor, and you have the retina back here. The retina is filled with photoreceptors that are able to essentially capture like light and such. But yeah, one interesting fact is your eyes are the one special organ that are actually part of your brain. Other ones are kind of separated from it, and you can imagine them like they have to transfer information through these pathways, while your eyes are directly connected to your brain. So yeah, there's something special there. And what's interesting about it, I guess, is that your eyes also have their own unique blood-brain barriers, which means, well, as some of the quirks of metabolism are the same as the CNS. In addition to the retina, uh, the eye also has a lot of accessory pathways, such as your pupil and your eyelid, iris, some other stuff, some physical protection like eyelashes, your eyelids and stuff, as I said. And your lenses accommodate light. They can bring to a focal point that by controlling the shape of the lenses using ciliary bodies. I should label that. Ciliary bodies. And retina, the retina down here is a sheet of neurons. There's a lot of photoreceptors, neurons, farthest away from incoming light. And photoreceptors only like they only take in oh yeah, in the order of transduction, you go photoreceptor to bipolar cells and the retinal ganglion cells. And photoreceptors only receive light coming from a specific point in visual space. So if the light is coming from this way, then you're gonna get it detected from here and whatnot. Another important feature is the macula and the optic disc. So imagine all the blood vessels and stuff, everything has to kind of connect to one thing to the brain. Well, this is the optic disc for axons and blood vessels. Now, because there's the optic disc, this means we have a blind spot, meaning that there's no photoreceptors here. We can't see anything here because this is where everything connects. And you also have a macula, which is where you see most of the color. So I guess we can talk about it, but photoreceptors, there's two types. One is called rods and one is called cones. The macula has a lot of cones, and this makes it such that you're able to see most color there. And photoreceptors on the other side, it's just kind of to see how it is. And these are all by opsin proteins. So yeah, all of them have an outer segment of yeah, I should say photoreceptors have the protein they use is opsin proteins with retinol, which is a vitamin A derivative, very important. And when light hits it, retinol changes from transform, and activates opsin. Rods mediate light in low light, but have low acuity. This is one thing you should note. Cones have low sensitivity but high acuity. Okay, I guess I should say this. Rods, low, high sensitivity, low acuity. Cones, low sensitivity, high acuity. Meaning they're much more specific. Cones are in the fovea centralis, and there's three types of cones actually, I should say. S cones, L cones, and M cones, where L cones are red, S cones are blue, and M cones are green. And the acuity from part of the cones comes from the fact that you only have one cone per one bipolar neuron. Whereas for rods, you can have two rods per one bipolar neuron. So what happens is all photoreceptors are release neurotransmitter in the darkness. So in the dark, they all have neurotransmitter release. And once the light hits it, it becomes hyperpolarized, so no more neurotransmitter. And this happens by deactivating sodium channels. Um, let's see if I can tell you anything about the order. I guess the biggest thing is when light hits one of the receptors, retinol changes to transform, and this activates opsin. And I guess this basically just leads to, and the opsin prevents any sort of NA channel movement, any changes in NA channels, deactivates them. Mm. Then processing of encoded as information from a reversed in polarity. And then visual information goes to the primary visual cortex, B1. And there's also the sort of reverse topology, whereas the left eye encodes information to the right side of the brain, or and then the right and left eye encodes information to the left side of the brain. But it's a bit more complicated than that, although I guess you shouldn't go too much in detail. Um, yeah, hearing is also another thing we should talk about. Detection of sound, you have the outer ear, you have the inner ear, middle ear, I should say, and then you have the inner ear. So the outer ear is only the external meatus acutus. The inner ear is filled with ossicles and the tympanum, and the inner ear is the cochlea. So the ossicles of the middle ear, ossicles creates vibration in the fluid of the inner ear of the cochlea. So what happens is the ossicles push fluid with the oval window towards the round window of the inner ear. The ossicles, okay, I should say ossicles push fluid of oval window of the inner ear towards the uh, round window of the inner ear. And this vibrates the inner ear, the semicircular canals, and the inner ear, and moves hair cells. Upon moving the hair cells, this creates the idea of sound. This displaces hair cells. You can also have the sense of equilibrium within the ear with the semicircular canals. This is located in the cochlea. And the semicircular canals, they kind of just vibrate and move according to gravity. And with that, they allow you to feel a sense of movement. And hair cells, let's talk about hair cells. So these are they have a kinocilium and a stereocilium. Moving in the direction of stereo inhibits hair cells, but in kino, which I guess is movement, stimulates hair cells. And within the stimulation of hair cells, the movement of hair cells relative to the tectoral membrane leads to the opening of mechanically gated ion channels, depolarizing them, and hence leading to sensations to the brain. And with this, the basillary membrane also differs in terms of flexibility. Actually, I should probably draw it out better. Let me see. So you have the cochlear duct, which I refer to as part of the round window and the oval window. The fluid enters the ossicles, what pushes sound waves hit the reach the ossicles, the tymphanium, which vibrate the ossicles and push fluid. I should say tymphanium pushes an ossicles, and vibrations moves fluid from oval window to round window. And moving through it, it affects the cochlear, it displaces hair cells in the cochlear duct, displaces hair cells in cochlear duct. And displacing these hair cells uh, relative to the tectorial membrane leads to action potentials that happen. I should get a new piece of paper. Polarized hair cells. Well, it activates voltage-gated calcium channels, which leads to depolarization. And the basal membrane is flexible, such that different frequencies will occur in different parts of the membrane. And the cochlear maps pitch, not space. So location of sound of an object is kind of interesting in a sense, right? Because there's going to be a difference. Let's say you are in this location and then you hear a sound from here. You can enter this ear. Let's say it's closer to the left ear than the right ear. And it takes a longer distance. So this would be an interval time difference. You might understand that one sound takes longer to reach one ear than the other. And you can also understand an interval level difference, which creates a sound shadow for the further ear. So what does this look like? So let's say you have a left ear and a right ear and you have amplitude on both sides. An interval time difference might mean that you'll hear this sound first on the left side before you'll hear on the right side. But level difference would mean that, oh, I will, this part will be much more intensive on the right side and it will be on the left side. And it reaches the primary auditory cortex of the hindbrain before reaching the forebrain. So yeah. Lastly, I guess we can talk about the enteric nervous system and some stuff about smooth muscles and stuff. So first, smooth muscle is found all over the body, and contractions in smooth muscle rely on thick and also rely on thick and thin filaments, but not in sarcomeres. So smooth muscle, thick plus thin, no sarcomeres. Smooth muscle also has two types, multi-unit or single unit. And in multi-unit, you use synapses. Single unit, use gap junctions and pace setter cells. Uh, Excitation contraction is also calcium dependent, but calcium is different. So first you have cal desmin relative to troponin. And so instead of revealing attaching to troponin, you attach to cal desmin. And well, after that, it's still the same sort of myosin head activation. Autonomic nervous system also comprises of efferent pathways, lapsing to smooth muscle in lungs, in adipocytes, in whatever sort of else it has visceral effectors. Uh, in the PNS, peripheral nervous system, somatic efferent pathways are usually monosynaptic, whereas just one synapse, whereas the autonomic nervous system are usually disynaptic, whereas it has two synapses. So this is pretty interesting. Well, the two synapses, one is called preganglionic, and this is one is called postganglionic. So within the digestive system, the efferent neuron systems, the ANS efferent neurons often synapse onto neurons of the ENS rather than directly onto the effector cells. And versatile effectors use metabotropic neurotransmitter receptors, which the key thing is that they're longer lasting. They can be stimulatory or inhibitory to the same neurotransmitter, and also diverse biochemical effects. Now, there's two pathways of the ANS we should note, the parasympathetic and the sympathetic. Now, the sympathetic, you can understand it as there's always a saying, a fight or flight and rest or digest, and this kind of makes sense. For sympathetic, you're generally seeing more fight or flight activities, but right, rest or digest is going to be associated to parasympathetic. And sometimes they're not always antagonistic, though. They're not always one thing works against the other. For example, both parasympathetic and sympathetic neurons help for orgasmic and ejaculatory reflexes in the reproductive system. And the sympathetic ganglia, what's interesting is that they're connected into a chain called the sympathetic chain, which means they can all be activated at once. All active. However, the preganglionic neurons of the two, and well, yeah, there's still this in the chain, and it's from T1 to L2. And then the parasympathetic is from cranial nerves 3, 7, 9, and 10, and then sacral nerves 2 to 4. These are the location of the preganglionic neurons. The relative length is also different. Sympathetic has a very short preganglionic. Well, yeah, they have a very short preganglion neuron, and then once they have a sympathetic ganglion, and then they have a very long postganglionic neuron. Parasympathetic, sympathetic, parasympathetic, they have a very long preganglionic neuron, and then they have a very short postganglionic neuron. What's also key is that the sympathetic neuron, well, for preganglionic neurons, they all use acetylcholine. But for sympathetic, you use norepinephrine, whereas for parasympathetic, you still use acetylcholine. Now, both neurotransmitters can activate a different set of receptors that can contain many different effects. The same neurotransmitter can be excitatory or inhibitory depending on the subset of the proteins expressed by the effector protein. For example, I'm not sure. Well, yeah, I guess it's just that. Visceral functions are controlled by visual reflexes that often don't include the CNS. For example, there's something called a long reflex, where stimulus goes to the CNS and then has a preganglionic neuron and then a postganglionic neuron. So this is a long reflex. So there's also a short reflex where the stimulus just finds an autonomic ganglion. This is a preganglionic and then postganglionic. So short has no CNS. And one example of well, I guess there's many examples of long and short reflexes. One example of a long reflex is for pupils in response to bright light constrict. An example of a short one would be gastroenteric reflex, which is your stomach filling increases intestinal motility. So yeah, this is going to be most of the content associated with the nervous system, and I hope you enjoy.